Hello, my name is Alex Martin. In June 2022, I graduated with a master's degree at the University of Kansas, where my research focused on aquatic botany, systematics, taxonomy, and aquatic ecology. I recently published a paper in the Journal of Aquatic Botany that looked at assessing taxonomy and systematics of the aquatic plant genus Vallisneria. This group is relatively understudied when it comes to overall species diversity and phylogenetics, despite them being very common in the aquatic plant trade. The first paper to assess taxonomy was published in 1982 by Richard Loden. His analysis of morphological characters resulted in a decision to reduce diversity within the genus to include only 2 species. In 2008, Donald Less and co-authors produced the first phylogeny for Vallisneria using molecular and morphological data, which resulted in a more speciose genus to include 14 species at the time. My research aimed to verify species diversity and relatedness, describe a new species from Northern Territory, Australia, and to address the natural and artificial hybrids that were being identified in both nature and within the aquatic plant trade. Unfortunately, I was unable to include 2 species from China into the study because I started this graduate project at the beginning of a global pandemic. Despite many restrictions, I was able to piece together a pretty fun project. One of my goals was to produce an updated phylogeny for Vallisneria, so I used gene sequences from GenBank along with new sequences from plants I obtained through both field collections and cultivated specimens. I also measured morphological features, as all the plants that I used for my study were grown in a common garden environment. My results supported the dissolution of 2 synonymies, and I also formally described a new species called Vallisneria jacobsii. By assessing sequence data and morphological features, it appears that Vallisneria plants readily hybridize. While I was on a trip to Central Florida to collect plants, I came across a population of Vallisneria in the Crystal River and in Kings Bay. They were planted there as a result of a restoration project. Sequence data suggests that these plants are the same non-native hybrids already documented by researchers in both Japan and Florida. It appears that this restoration project accidentally collected these non-native plants thinking that they were native to Florida, and then grew them in mass in greenhouses and then replanted them within the entirety of the bay. And what's important here is more and more organizations become involved in restoration ecology of aquatic ecosystems, it becomes very important that we understand species diversity within these project areas. So while the Vallisneria that were planted in the Crystal River provide a great food source for the migrating manatees that visit the bay every winter, the intentional planting of non-native hybrids can potentially become an invasive species issue later. The accidental spread of non-native plants can easily be avoided with a simple DNA test. So my advice for anyone interested in aquatic botany is to use citizen science applications like iNaturalist, as they're incredibly useful for tracking distribution and morphological features from living plants. Herbarium specimens are useful for many plants within botany, but unfortunately they aren't great for Vallisneria. Many of the features I was interested in are lost when these plants are dried and pressed, so being able to see living plants was very important. The project that I completed left a lot of unanswered questions, so I'm hoping that some future researcher will take the reins and answer some of these questions later on. So I thank you for your time.